Okay, thank you, Pascal. And thank you for inviting us to present our work again here in Basel. As Pascal mentioned, now we enter a little bit more in the experimental part of nanoparticle design. And in the case of malaria, is well known that actually nanomedicine, it cannot be that image represented on the upper left because that is simply too expensive. We cannot, besides being science fiction, we always must take into account the economic landscape when designing new medicines for malaria, especially if they are nanomedicines. Then, but we have the tools. We have nanoparticles and polymeric nanoparticles and liposomes that are working very well and which are not science fiction. However, it's true that there is a wealth distribution inequality in the sense that there is overwhelmingly more funding for diseases like cancer or neurological disorders than for infectious disease. And this is not proportional to the number of people in the world who are affected by both. Which means that whatever we design, especially if it's nanomedicine, it has to be economically affordable. And in the case of malaria, this is not so easy. This is the classical scheme of a red blood cell infected by the malaria parasite. And after so many years of evolution with its human host, Plasmodium has developed very tricky strategies to escape from the immune system and from any drug that we have been dumping into it for the last 100 years. Essentially, it hides inside the red blood cell without exporting too many proteins until relatively late in its blood cycle. And it's also hidden behind a parasitophorous vacuole. And you also have, obviously, the very parasite plasma membrane. And if you want to target some of the internal organelles, like the apicoplast, which is one of the most interesting targets, you would have to go through 4 different membranes. And then this would require consequently complicated nanovectors. This is what some people have called the Russian doll strategy, because you encapsulate your drug in a first nanovector, then in another one, until you reach your target, if this is deep inside the parasite. We must simply forget about this strategy, because we must go to things which are easier, and essentially I doubt that for malaria we can think of something that it's more complicated than simply encapsulating the drug in a nanoparticle like a liposome or some type of polymeric nanoparticle. So we have to be pragmatic and acknowledge that we must work with simpler systems. Then one of the most simple systems is obviously simply encapsulating the drug, for instance, in a liposome. Then when you do this, you generally obtain an improvement in drug efficacy. We have been trying with several different systems, and consistently you can achieve more or less 1 order of magnitude improvement in drug efficacy. You can improve the efficacy of drugs about 10 times. But this is not enough. We need clearly to go beyond that. And then what we think that we can do, and this is the aim of the main part of our work, is starting from this original relatively simple nanovector, improving its performance by changing some of the things that it contains, either the drug, the encapsulating structure, or the targeting molecule, which are the 3 essential elements of any nanovector, I think. Is what we call the Lego strategy. We have a good starting point, and now we simply have to change these different elements in order to improve it. And here is where I will present in the next slide some attempts that we are doing to new strategies of combination strategies against malaria. Combination, until now, is understood, as Reto said, as combining two drugs. And in this way, you clearly can improve efficacy, and essentially you can limit the evolution of resistance because you kill the parasites faster. But probably this combination strategy, which started not so long ago, in the midterm we will see also resistance to this, because simply it's two different drugs acting on different targets, but they are nevertheless the classical antimalarial drugs. Then we think that maybe it could be explored a combination of a drug. This is something that any nanovector will obviously contain, but some other property that we can impart to the nanoparticle, and which is not simply a drug, and that maybe in some cases it not target the parasite in a single gene product. You will see more or less what I mean with the next slide. For instance, our first combination strategy, all of them are under study now, so I cannot offer definitive results about any of them. So these are simply 4 or 5 ideas that I throw to the audience. One first combination strategy stemmed from our observation that when we were using liposomes, and those liposomes contain some fluorescent lipid, as you can see here, the blue DAPI staining tells you which of these red blood cells contain plasmodium. And then as you can see, the fluorescence of the lipid that went with the liposome could be targeted, actually, because we know the way to target these liposomes with antibodies or heparin or some other molecule. And not only was targeted to the infected erythrocyte, but it penetrated the red blood cell, and actually it localized with the plasma membrane of plasmodium itself. So somehow the lipids in a liposome are capable of being soaked up by the parasite, which led us to the proposal, especially after seeing that some of the lipids that we were using, particularly the one that we had to employ to target, to bind the antibodies to the liposomes, this lipid is very toxic for plasmodium. We realized that when we increased the proportion of this lipid in the liposome, the parasite was dying without any need for incorporating a drug. Then what we are proposing is that we might probably make our liposomes with a drug that will have its expected activity, but then also it would be like incorporating a second type of drug, in this case a lipid, but that its primary role would be like making the box of a present. So you give the present to plasmodium, it eats the drug, but then the box can still be used. It has like a second life. This would possibly reduce the cost of development, because by developing a single nanovector, you are incorporating there 2 different activities. A second one that we explained last year, so I will be very fast in reminding you this, is that this is the classical way to deliver antimalarial drugs, where you target your nanoparticles to the infected cells with the idea of killing the parasite, but then those parasites that escape, they will have plenty of red blood cells to invade and survive. Then our new approach consisted of targeting the drug-carrying immunoliposomes to all red blood cells by choosing an antigen like glycophorin A that is in all erythrocytes. We were also loading drugs in non-infected red blood cell. As long as this drug doesn't interfere with the normal red blood cell metabolism, which for most of the antimalarial drugs I think they don't, what we were achieving here, it was a very good efficacy because we think that plasmodium, when it enters the red blood cell, if it encounters already the drug from the beginning, it's very complicated for the parasite to evolve and to live. This is what we call the Little Red Hood strategy, because when you enter your home already, the wolf is there. And in this situation, as you can see here, we could finally completely cure the animals and we could eliminate the parasitemia. So this is, I think, one of our best beds for development. A variation of this that we have recently implemented is you can use also antibodies that besides targeting your target cell have some other physical activity, like related to rosetting, which is the property of infected red blood cells to bind non-infected red blood cells and make these aggregates that are very dangerous because they clog the little capillaries, and they are one of the main problems in cerebral malaria. If you use for your targeted liposome an antibody that recognizes some of the antigens, like some PFE-MP1 epitopes that are involved in rosetting, besides killing the parasite with the drug that the liposomes contain, you could also significantly reduce rosetting in the system, which is also a very good news. Besides liposomes, we also have polymeric nanoparticles. Also, we presented this data a couple of years ago, so I will also go quickly through them because actually we realized a new thing about these polyamidamine polymeric nanoparticles. They are capable of encapsulating drug, and the surprise was that they do not need any targeting antibody to reach infected red blood cells because somehow they have a particular tropism towards this type of cell. They are capable of recognizing them specifically and not non-infected red blood cells and of penetrating them. They have a quite good drug release over 24 hours. They work very nicely in vitro and also in vivo. The thing is that these very good in vivo results, this is chloroquine, which as you know is one of the best antimalarial drugs, and then at a concentration that chloroquine was incapable of curing the animals, encapsulation of chloroquine in these polyamidamines was able to cure the mice. And actually, the result was too good for what we expected. So it could not be explained simply by a good targeting. And then we suspected that here we had some overlapping activity. And then when we looked more carefully at the cultures, we realized that very often the cultures treated with these polymers, the parasites there were not able to invade red blood cells. So there was an invasion inhibition that we think it's something that it's incorporated in the system besides drug activity. And then when we look more closely into this, indeed, polyamidamines, here fluorescently labeled, they bind very strongly to the parasite itself, to the merozoite when it's free, like here, that these merozoites are free because the red blood cell broke up. And also, as we have seen before, these polyamidamines are capable of entering clearly the cell. So they enter and they bind, and here is where they might be, this coating of the merozoite probably is what it's making difficult for the cell to invade new erythrocytes because the molecular machinery is sterically impeded. And this is probably a good example of a Trojan horse because the polyamidamines don't do essentially anything to the parasite. The parasite sees that the polyamidamine is coating it, it doesn't kill it, but then it realizes that when it exits the cell, it cannot invade. So it would be a type of new activity, again, that we are adding. But here the problem is that polyamidamines by themselves have some unspecific cytotoxicity. In addition to the antimalarial effect, which you can see here with growing concentrations and size of the polymers, when you do your test with endothelial cells, you also see some residual activity. And here is where we were then exploring other molecules like heparin, which has also a good targeting, as we could see in the past. And then we explore heparin as a substitute for antibodies in targeting strategies. And the idea is that if you load a liposome with a drug and target it with heparin, heparin would have a double activity by targeting the liposome to the target cell, but also afterwards it would express its own antimalarial activity, as we could clearly see in this kind of assays. When free drug has a relatively poor activity, here is primaquine. Empty liposomes with heparin have a significant activity, which comes actually from heparin, because here there is no drug. And when you combine both things, drug plus heparin targeting the liposomes, we have an increased activity that cannot, it's not simply the addition of both activities individually. So there is some type of synergistic effect. So heparin has a double life, but the problem is that it's a potent anticoagulant. And then this is my last slide. We are exploring other molecules similar to heparin, but with low anticoagulant activity. And here is where, in collaboration with a group from Brazil, we have been exploring some sulfated polysaccharides of marine origin from sponges, sea cucumbers, and algae. They work very nicely, some of them. In addition, we could see that the animals treated with those polysaccharides, they had antibodies against Plasmodium proteins in the plasma, and most interestingly, those animals, when reinfected with malaria and not treated, they were cured. So here there is clearly some protective effect of heparin, probably derived again from this invasion in the inhibition that keeps the parasite in the blood circulation for a longer time, and then the immune system has time to build an immune response. So the conclusion is that we think it doesn't matter if we talk of Lego, magic bullets, Russian dolls, little red hoods, or Trojan horses, that nanomedicine can be an option for diseases of poverty. And I thank all the people in the group and our funding agencies, and I thank you for your attention. Thank you very much.
Thank you very much. Time for one question. I haven't quite understood what antibody do you use to target the erythrocytes? Or the... In
the beginning, we used a commercial antibody that it was described as binding infected red blood cells, but then because the efficacy was not very good of nanovectors, we looked at which antigen was targeted, and it was actually an intracellular antigen, a Maurer-Cleft-associated protein, but that in about 5% of the cases it was exposed extracellularly. And then the other model, for that we use glycophorin A just as a universal target for red blood cells.
Yeah, malaria parasite is known to take up to 2 weeks in the liver stage before it comes out in the bloodstream. At that point, you are not sick yet. Are you trying, or are you thinking of ways of actually curtailing its life in the liver stage, kind of a prophylactic treatment, so that one does not need to be sick, especially for pregnant mothers or people who are HIV infected, or people who are traveling. Is this one of the ways you're thinking of intervening?
Yeah, this is one of the main targets that the malaria community is trying to solve, this targeting of parasites in the liver stage. It's not easy, I think. We didn't never work with that stage. It's not easy because there are very few hepatocytes, I think, that are infected by the parasite, and it's very hard to get to them. But it has been described that, for instance, heparin is one of the receptors for the parasite in hepatocytes, and then we are planning maybe for future projects to use some our heparin targeting that we have learned to try to see if liver stages can be targeted with nanoparticles. But I think one of the most prominent groups working on that is in Lisbon, at the University of Lisbon. Miguel Prudencio, I think. They are working very intensively with targeting of liver stages. So they are progressing very quickly. I think in a few years we will see something along that line.
Just another one.
No, we have to continue. I'm sorry.
Yeah, a quick one. Pregnant mothers, they normally keep on getting malaria, and it has been realized that the parasite hides in the placenta. And unless it's tackled in the placenta, they just keep on getting sick all the time. Are you also thinking of how to intervene on that one as well?
Yes, for the placenta we have a relatively good target, which is chondroitin sulfate, which is a relative of heparin. And the parasites that bind to the placenta, the receptor for there is chondroitin sulfate. Here we are working with some Plasmodium strain that is binding to this receptor. And here we are working on that. Yeah, I can tell you that we are trying to develop some nanovectors for placental malaria.